Topic of my presentation is preparation, optimization, characterization, and in vitro lipolysis permeation of self-nanoemulsifying drug delivery system containing linagliptin and quercetin. Firstly, we will move ahead with introduction. Linagliptin on oral administration suffers from two major issues. First, it shows only 30% of oral bioavailability, that is due to high first-pass metabolism, and this problem can be tackled using self-nanoemulsifying drug delivery system. We show the lymphatic absorption with the help of this chylomicron, which is present in enterocyte, which take drug into the lymph vessel and which is ultimately reached to blood, and this way it can bypass the liver. The second problem of low permeation due to PGP efflux, this can be overcome by quercetin. Quercetin being a PGP efflux inhibitor, it blocks the PGP, and this way we can improve permeation and oral bioavailability of the drug linagliptin. These are the four objectives we will be going through: screening of oil, surfactant and co-surfactant, then optimization, then characterization of formulation, then integrated in vitro lipolysis and permeation study. So moving with our first objective, screening of oil, surfactant and co-surfactant. Both the drug was showing maximum solubility in capmol MCM C8 out of selected various oil. So this first box shows the screening of our surfactant, and second box shows the screening of co-surfactant. Out of the various surfactant, we selected Cremapur RH40 based on their emulsification ability to oil capnul MCM C8. This emulsification ability was judged based on the droplet size, PDI, and percentage transmitters. The same thing happened did with co-surfactant screening. Labrafil M1944CS selected in the same manner as a co-surfactant. Then we did optimization, where we optimize various factors, and these are the response surface curve obtained through optimization for size, PDI, and transmittance. We show the significant interaction between factors. This is a formula, optimized formula we obtain at the end of optimization. Then we did the characterization of our optimized formula. Firstly, we'll characterize it for size and PDI. Size was found to be 45 nanometer, and zeta potential was found to be minus 11.49 nanometer, which was in a good range. Then we did morphology. Then we check morphology of constituted snails using transmission electron microscopy. The particle was found to be very spherical. Globules were found to be spherical and within the size as suggested by Malone zeta sizer. Then we did robustness to dilution test, where we diluted our formulation to 200, 400, 600, and 800 times. Then the particles, as we can see in the graph, the particle size were increasing as we are diluting more and more. But at the maximum dilution, even at the maximum dilution, the particle size was 175.5 nanometer, which was still in nano range. So the robustness dilution was accepted. Then stability studies in simulated gastrointestinal fluid. We took water, HGF and SIF. After 2 hours, the particle size were same and PDI were also same. But the only change happened in simulated intestinal fluid PDI in both after 6 hours and 2 hours. Then we did characterization for a heating cooling cycle and freeze-thaw cycle. In heating cooling cycle, we kept our sample at 4 degrees Celsius for 48 hours, then 45 degrees Celsius for again next 48 hours. We repeat this cycle for 3 times, and after the third cycle, the particle size was found to be 50 nanometer with the narrow PDI, and thus after freeze-thaw cycle, the particle size obtained was 87.58 nanometer with a slightly higher PDI. Even though the particle size distribution got disturbed, the particle size was still in nano range, and the color was emulsion turned to slight red, slight yellow. Then the main part of this project, integrated in vitro lipolysis and permeation. We use this PS stat auto titrator for lipolysis experiment, where we fill it with 0.5 normal NaOH. In this vessel, we fill digestive media which contain our formulation. We know that our formulation is lipid based, and lipid is nothing but glycerol molecule attached with 3 fatty acids. When this came in contact with the enzyme porcine pancreatin, it breaks the lipid molecule into glycerol and fatty acid. This enzyme is prepared freshly before experiment. This free fatty acid release decreases the pH of media, and which is sensed by the pH electrode deep into it, and the instrument add 0.5 normal NaOH to compensate the decrease in pH due to free fatty acid, and this amount of NaOH is considered as a directly proportional to the amount of digestion, and the NaOH which combined with this free fatty acid and form the palette phase, and this is the NaOH we use for our experiment. Then this is an experimental setup for a lipolysis experiment where digestive vessel, it contains digestive media and our formulation. Then it dipped with a autoburate and a pH electrode which is attached to the instrument further. We take samples at every 15 minutes of lipolysis. After taking sample, we need to stop digestion at that particular time point, and for this we use a chemical called 4-bromophenyl boronic acid, and this stop the digestion at that particular time point. After centrifuging it to 13,300 rpm, it forms the two phases. One is aqueous phase and other is palette phase, and this aqueous and palette phase is drug is determined in these phases using develop HPLC method. Then these are the results obtained by lipolysis experiment, where in first box is for linagliptin. We can see the maximum portion around 80% was found in aqueous phase, which is ultimately will be available for absorption. And we can see as the time passes, the drug was showing sign of precipitation, so the drug is getting found in palette phase. And the second box for quercetin, we can see the quercetin present around approximately 50% in aqueous and in palette phase. This is because the quercetin was showing the sign of precipitation, and this precipitated quercetin is precipitated in amorphous form, which is concluded through its good permeation result, because in amorphous precipitate form, it was in equilibrium with aqueous phase. Then this is a setup for permission study, where we take sample different time point sample of lipolysis. We put them in 5 different transdiffusion cell for permeation. This donor compartment can contain lipolysis sample, which includes which have itself oil, then micelle, then digestive product. Drug get permeated through this cellophane membrane, and we quantify the drug at various time using develop HPLC method at which we have taken at various time point, and this data can be further used for IV-IVC correlation. Then these are the result obtained through permeation study. 0 time point sample of lipolysis, when we put for permeation, it show a very good permeation. Within 15 minutes 100% of drug got permeated, and whatever the remaining above 100% is cumulative of whatever drug already present in acceptor compartment. So the quercetin was also showing good permeation result, but as the time was passing and enzyme was acting on formulation, the permeation profile was getting disturbed, or not even 100% drug was able to permeate. So we can conclude that lipolysis have a profound impact on our formulation, which is interfering with permeation process. We can see the time profile, permeation profile of 30-minute sample of lipolysis when put for permeation, same for 45 minutes. The quercetin was showing good permeation in all the time point. And this is a 60-minute sample of lipolysis when put for permeation, show this kind of permeation profile, and quercetin shows the 100% permeation again. And conclusion, Capmul MCMC8 Cremophor RH40 Labrapil EM1944CS forms a good nanoemulsion containing linagliptin and quercetin. In characterization, formulation was found to be stable, robust, and optimum. Lipolysis have significant impact on formulation. Permeation was good in initial stages of stages of enzyme presence, future perspective, pharmacokinetic or animal studies. Thank you so much.